Hello dear students, welcome to the lecture on the topic Evolution and Diversity of Angiosperms. In this lecture, we will be discussing number first, the diagnostic features of angiosperms. Second, we will try to understand the evolution in angiosperms. And third, we will try to highlight the diversity in angiosperms. So, starting with the diagnostic features of angiosperms. The angiosperms, sometimes also called as flowering plants or magnoliophyta, or a name given to these angiosperms is anthophyta. This is a group of plant which is most common and diverse group of land plants with over 2.6 lakh species distributed all over the world. These angiosperms grow virtually in every habitable region and are dominant in some aquatic and most of the terrestrial ecosystems. In fact, majority of our plants which we use economically for different purposes like food, fodder, fuel, fiber, or even medicinal plants. Majority of these plants belong to angiosperms. At present, the angiosperms are thought to be very closely related to another group of plants called as gymnosperms. That's why sometimes we call angiosperms as sister group to the gymnosperms. Gymnosperms are basically plants with nacked seeds. There are several diagnostic features, unique features, which distinguish these angiosperms from rest of the land plants. Out of the various diagnostic features, the most important is the presence of flower within the angiosperms. The flower is usually associated with accessory whorls in the form of perianth. In a flower, we have also the reproductive parts in the form of androecium and gynoecium. Androecium is comprised of the stamens, that is the male part of the flower, with two lateral theca. Each of these theca is comprised of two microsporangia. We have a reduced 3-nucleate male gametophyte within the angiosperms. The female part, that is the carpellus within a flower, it has a stigmatic surface which is used for pollen germination. One more diagnostic character of angiosperms is that they have a reduced female gametophyte, which usually comprises of 8 nuclei but 7 cells. The seed which is produced within the carpellus, it is enveloped, it is surrounded within the fruit wall. One more unique feature associated with angiosperms is a phenomenon called as double fertilization. What is this double fertilization? It leads to the formation of a triploid nutritive tissue called as endosperm. Also, we have, anatomically speaking in angiosperms, the presence of vessels in the xylem tissue. One more diagnostic feature of angiosperms by which it differs from rest of the land plants is that their sieve tube elements are always accompanied by one or more companion cells in the phloem tissue. After discussing these diagnostic features, which will help us in understanding what basically these angiosperms are, now let us take the evolution of angiosperms in much more detail. Charles Darwin, an evolutionary biologist, once remarked that the relatively rapid evolutionary diversification of angiosperms, he used a phrase, abominable mystery. The earliest fossil record of Angiosperms is in the form of pollen grains that has been recorded from the earliest Cretaceous period. It is a geological period round about 140 million years ago. That means we have the first record of evolution of Angiosperms by the way of their pollens about 140 million years ago. The flower, which is most distinguishing feature of Angiosperms, it dates back slightly later in the fossil record. That is about 130 million years ago. These earliest flowering plants in the fossil record can now easily be assigned to the present day living groups of Angiosperms. Many major Angiosperm lineages can be recognized by the mid-Cretaceous period. For example, water lilies, magnolids, and the eudicots were present by 125 million years ago. Traditionally, the closest relative of Angiosperms that I have already mentioned were the Gymnosperms. They have been considered as the closest relative of Angiosperms. However, the details of Angiosperm evolution from a possible Gymnosperm ancestor is not very much clear. There are various hypotheses that trace the evolution of Angiosperms from Gymnosperms. One of the widely debated hypotheses is that Mesozoic bennettite tails, also called as cycadoids, could be the possible ancestors to Angiosperms because these cycadoids, they produced large flower-like reproductive structures. These flower-like reproductive structures have pollen-producing organs which are surrounded by a central stalk which bears naked seeds. Gymnosperms such as T-tails along with the already extinct bennettite tails were widely believed to be related to the Angiosperms on the basis of several morphological similarities. Some of these morphological similarities are vessels in the wood. Both Angiosperms as well as knee-tails and the bennettite tails, they have vessels in their wood. One more similarity is reticulate leaves in Neetum and also the flower-like reproductive organs. Based on the phylogenetic analysis of morphological data, it was concluded that Angiosperm formed a clade, formed a monophyletic group with knee-tails on one side and bennettite tails on other side. This group, both Angiosperms and knee-tails and bennettite tails together, they were referred to as anthophytes. This name anthophyte was in relation to the flower-like reproductive structures. However, very recently, this viewpoint has completely changed. For example, vessel elements are now reinterpreted as being derived independently. That means a product of parallel evolution. For example, in knee-tails, they are derived from circular bordered pits, whereas in Angiosperms, they are derived from tracheids with scalariform pits. One more widely debated hypothesis about the evolution of Angiosperms is that Angiosperms were derived from an extinct Mesozoic pteridosperms. The pteridosperms, also sometimes called as seed ferns, these were a group of Mesozoic fossil plants that possessed seeds. They had seeds, but their leaves were just like ferns. Some of the members in the pteridosperms may represent possible angiosperm progenitors. One such fossil taxon that best exemplifies a possible transition to angiosperms is named as Cattonia. This Cattonia possessed reproductive structures very similar to those what we see in angiosperms. In Cattonia, the male reproductive structure, it resembles anthers in consisting of a fusion product called as synangium, which bears 3 or 4 microsporangia. However, these synangia differ from angiosperm anthers in having radial symmetry in contrast to bilateral symmetry in majority of the angiosperms. The female reproductive structures of Cattonia consist of a spike-like arrangement of units that have been termed as cupules. Each cupule encloses a cluster of unitasmic ovules or seeds with a small opening in the cupule near the proximal end. The cupule has been hypothesized as being homologous with the angiosperm carpel. However, it differs in having a conduplicate megasporophyll which bears ovules along its 2 margins. Additionally, the monosulcate pollen grains have been reported at the micropyle of Cattonia ovules. The latter is a strong evidence that the pollen grains were transported directly to the ovules, perhaps by means of a pollination droplet, as we commonly see in present-day gymnosperms, rather than what we see in angiosperms as a stigmatic region, where pollen tubes are formed in the angiosperms. Therefore, the cupule did not function as a carpel in terms of a site for pollen germination, what we see in angiosperms. Another viewpoint about the cupule of Cattonia is that it is a homologue of second integument which is present in the ovules of majority of the angiosperms and might have evolved by reduction of the number of ovules within the cupule to just single. Although the homology of reproductive structures in Cattonia with reference to angiosperms is largely unclear, still they are closely related to the angiosperms than to the gymnosperms. Very recently, a fossil plant by the name of Archiphractus, which was collected from China, is believed to shed light on the early evolution of angiosperms. The fossil taxa dates back to as early as 130 million years ago during the early Cretaceous. The genus Archiphractus, now with 2 described species, was apparently an aquatic plant and had dissected leaves and elongate reproductive axes. Each of these reproductive axes possess paired stamens below and several seeded carpels above. The interpretation for the relationships of these structures with the present-day angiosperms is highly debatable. According to one hypothesis, the reproductive axis that we see in Archiphractus is an entire perianth-less flower with stamens below and carpels above. The axis is perhaps homologous to an elongate receptacle much similar to the magnolites. Thus, the reproductive structure in Archiphractus might represent an ancestral flower, or we can say a flower precursor, and therefore this Archiphractus might be sister to all the present-day angiosperms. According to an alternate hypothesis, as I already mentioned, that this is highly debatable. That's why there have been different hypotheses. According to an alternative hypothesis, this reproductive axis of Orchifractus is not a single typical flower but an inflorescence. Inflorescence means an aggregation of flowers wherein we have reduced male and female flowers, as we see in many of the aquatic angiosperms today. From this alternative viewpoint, we can say the Orchifractus may represent an extinct offshoot of an extant lineage. That means which has been already extinct but an offshoot of a living lineage within the angiosperms. There are many guesses that it can be the extant lineage can be nymphales or some other aquatic angiosperm. After discussing these various hypotheses, there are various evolutionary novelties that we see in angiosperms that are believed to have played a pivotal role in the diversification of angiosperms. Out of these various evolutionary novelties, the pollination and fruit dispersal mechanisms are of great importance. The pollination, in particular by insects, what is called as anemophily, is even known from the fossil bennettite tails and seed ferns. The insect pollination was established by the time the core angiosperm had already evolved. It was probably first carried out by pollen-eating or pollen-collecting insects, especially beetles and flies. And the flowers were pollinated by nectar-collecting insects, which evolved much later in the geological time scale. It is now abundantly clear that the diversification within some angiosperms, for example the orchids, and the insect lineages on the other side are intimately linked together in a co-evolutionary manner. In angiosperms, we see the variation in fruit morphology is largely related to the use of different dispersal agents. The fossil fruits and seeds of angiosperms from the Cretaceous era are generally small and little specialized. Therefore, they were little specialized for dispersal by mammals or birds. Adaptations for dispersal by frugivorous or granivorous animals in most of the lineages of angiosperms originated in the tertiary period. The tertiary is the same period when the radiations of modern birds or mammals have occurred. Consequently, as a result of this, the evolution of large, bright colored fruits and seeds in majority of the angiosperms was linked to the evolution of these animal groups, which have helped in their fruit dispersal. After discussing these various evolutionary hypotheses and various novelties about the angiosperms, now let us discuss the diversity in angiosperms. All that whatever the evolution has taken place in angiosperms that have led to the diversification of angiosperms. Once angiosperms evolved, as we have already discussed, they diversified rapidly into several distinct lineages and gradually became the dominant components of the global vegetation. At present, the angiosperms are represented by about 260,000 species. We have at present the total number of land plants is 3 lakh species. Out of these 3 lakh species, angiosperm consists of 260,000 species. These angiospermic species, they display an amazing diversity of morphological, anatomical, biochemical and molecular characters. And these species of angiosperms, they can occur in an extremely wide array of terrestrial habitats. Or many angiosperms we see, they have secondarily their habitats as the aquatic ecosystems. Traditionally, the angiosperms were recognized under two main groups of monocotyledons and dicotyledons. However, such a separation nowadays is believed to be an arbitrary one, and it is no longer accepted as per the principle of monophyly. For instance, the possession of two cotyledons in the traditionally delimited dicotyledons is now considered to be an ancestral feature called as plesiomorphy and therefore paraphyletic. On the basis of recent kaleidostic analysis, the angiosperms are now recognized under the four so-called groups, not the erstwhile two groups, monocotyledons and dicotyledons, but four different groups. The first group is what they call as antegrade. Second is magnolids. Third is monocots, and the fourth is eudicots. Let us discuss these one by one. The first one is the antegrade. It's a small assemblage of non-monocot and non-eudicot angiosperms. That means they are outside of monocots and eudicots. This antegrade, it includes lineages such as embryolales, nymphiales, ostrobaliales. This group may be characterized by carpal margins usually sealed by a secretion, whereas we have seen in majority of the angiosperms the carpals are closed by postgenital fusion of epidermal layers. Members of these lineages, that is the antegrade, they have 4 nucleate female gametophyte and interestingly diploid endosperm, whereas we know most of the angiosperms have an 8 nucleate female gametophyte and a triploid endosperm. The second group that is presently recognized under the angiosperms is called as magnolids. It basically comprises rest of the non-monocot and non-eudicot angiosperms. It's a monophyletic group, that's why it's sometimes called as clade. This clade includes magnoliales, laurels, canalales, and pipirales. The third group is the commonly known monocots or monocotyledons. They have been long recognized as a major and a distinct group. The monocots comprise nearly 56,000 species. That is, it comes round about 22% of all the angiosperms. Monocots include the well-known aroids, arrowleaf plants, our common lilies, gingers, or the diverse orchids, irises, palms, and the most economically important, grass. Traditionally, the monocots have been distinguished by the occurrence of trimerous floral parts. That means the floral parts are in 3 or multiples of 3. However, recently, this feature, that means the trimerous, is now considered to be an ancestral condition, a plesiomorphy. This ancestral condition is present in several non-monocot lineages of angiosperms, such as laurels, magnoliales, and pipirales. All the members of the monocots, they are characterized by few apomorphies. That means some features which are common to all the monocots. These may be, number 1, sieve tube plastids with cunate. Cunate means wedge-shaped proteinaceous inclusions of P2 type. Second apomorphy is atactostele stem vasculature. What is this atactostele stem vasculature? It consists of numerous discrete vascular bundles. Third apomorphy is the parallel leaf feniation, and the fourth one and the most obvious one is the presence of a single cotyledon. The various orders that have been recognized under the monocots at present are ecorales, elismatales, mostly the aquatic herbs, aspergales, a large order wherein we have the Orchidaceae family, the orchids, a diverse group of organisms. Then we have dioscorales, Liliales, the common lily family we have in under liliales. Then ericales. Ericales are the common palms, coconut tree. These are the various palm species which have been grouped under the ericales. Then we have commelinales. Then also gingiberales. And lastly, the most economically important, poales. All these orders have been recognized under the monocots. The fourth group of angiosperms is called as eudicots. Eu means here the true dicots. It's a large assemblage of angiosperms which is characterized by many apomorphies. Apomorphies means the shear derived characters. The apomorphy in eudicots are: number one, the tricolpate pollen grain. Number second is floral parts, they are arranged in whorls, not spirally arranged. The perianth which is present in the flowers of eudicots, it is clearly distinguishable into calyx and corolla. And these eudicots have plastids of sieve elements with starch grains called as S-type. The eudicots is a large group. It comprises of about 24 orders. At present, eudicots comprise nearly 1,90,000 species. This comes about 75% of all the angiosperms are represented by the eudicots. The various orders that have been recognized under the eudicots include, starting with ranunculales. Then we have proteales, a group of plants which is mostly restricted to South Africa. Then we have caryophyllales, a large group of plants which is recognized under the order caryophyllales, followed by centellales. The centellales order is followed by saxifragales. Then geraniales. Geraniales is an order wherein we have the common geraniums which we use as ornamental plants. Then we have myrtales. Myrtales is the same order wherein we have the eucalyptus-like trees. Then followed by cucurbitales. It is an order wherein we have some plant taxa which we use in as common vegetables. Then followed by favales. Favales is an order wherein we have the legumes. Followed by phagales. This phagales order is followed by malpighiales, which is followed by oxylidales. In addition to these orders, we have few more orders in the eudicots like rosales, brassicales, malvales, sapendales, cornales, ericales, gentianales, lamiales. Lamiales is the common order wherein we have the mint plant. The various aromatic plants, they belong to the lamiales. Then we have solanaales, apiales, astrales. Astrales is a very large group of plants, eudicots, and this astrales is lastly followed by one order called as dipsacales. So dear students, with this we come to the end of today's lecture that was on the evolution and diversity of angiosperms. Hope you have enjoyed today's lecture. See you again with some another topic. Till then, goodbye.